Antoine Béchamp was studying the origins of microbial life by looking at fermentation. He found microorganisms in plant, animal and natural minerals that didn't come from the outside. These little organisms he called microzyma, and it was observed that they can develop and change shape. This is called pleomorphism. Béchamp realized that disease did not come from germs in the air invading from the outside. Disease was produced within the body due to an unfavorable internal environment brought about by poor nutrition, drugs, toxins and various other factors. Pasteur, on the other hand, viewed these entities as being in the air, outside the body, that there were specific invading microbes associated with specific diseases. Antoine Béchamp accused Louis Pasteur of taking his theories and distorting their meanings, thus steering medicine down a dead-end path. Pasteur took Béchamp's experiments to prove his theory, and since these invading microbes can go into a vessel and spoil beer, wine and milk, known as fermentation, they can enter our bodies and eat us from the inside, causing disease. According to Wikipedia, Louis Pasteur's pasteurization experiment, as it later became to be known, illustrates the fact that the spoilage of liquid was caused by particles in the air rather than the air itself. These experiments were important pieces of evidence supporting the germ theory of disease. But is this really what the experiments showed? If we look at the first experiment, any bacteria in the flask are killed by the heating process. Heating to high enough temperatures will denature all organisms. Experiments 2 and 3 show that bacteria will recolonize the nutrient broth after sterilization if they are able to reach it, which shows that bacteria are everywhere and will grow in nutrient broth. So how is this evidence for germ theory? Is it not evidence for terrain theory? The nutrient broth is not an organism such as a human. It does not get diseases. The experiments do not show that bacteria can invade a host. They show that bacteria will grow and multiply in suitable terrain. Wikipedia describes the liquid nutrients as being spoiled by the bacteria. But think about it. If you put out a suitable meal for your family and they eat it, are they spoiling it? In the same way, you have just provided a suitable meal for the bacteria. Pasteur did not anticipate that in the future there would be an electron microscope with a much higher magnification than the optical microscope that would allow us to see small structures that weren't visible before. Now let's cross over to Germany. Bacteriologist Robert Koch had discovered the common bacillus and proclaimed it to be the cause of Asiatic cholera. Dr. Klein, who was about to proceed to India to investigate the origin of that disease, did not believe in Professor Koch's statement and experimentally drank a wine glassful of common bacilli in pure culture. No effect followed. Robert Koch had developed postulates as a logical scientific way of proving germ theory. But these postulates worked against him and others trying to prove the germ theory. He knew after his 1884 experience with cholera that it was often impossible to induce a disease experimentally in animals. Instead of realizing that his postulates worked as designed by disproving microbes as causative agents of disease, he decided to amend the postulates so that invisible entities only had to be believed to be the causative agent of a disease. Instead of scientific proof, now you just needed faith. 'Cause I gotta have faith. I gotta have faith. Because I got to have faith. I got to have faith. The germ theorists distanced themselves from Koch's postulates because if they followed the scientific method, they ended up refuting their own theories. In 1937, Thomas M. Rivers, the director of the Rockefeller Institute for Medical Research, proposed modifications to the original Koch's postulates. These were called Rivers' postulates. You can see in the picture by the color coding that there is some overlap. One criteria not required by Rivers' postulates is the first one by Koch, that the microorganism is found in ill people but not healthy people. Rivers stated that, in the first place, it is not obligatory to demonstrate the presence of a virus in every case of the disease produced by it. But if the virus is not present, how can you say it caused the disease? Going back to Pasteur and Koch, why were these men who couldn't scientifically prove their theories put on pedestals and true scientists ignored? They were used by the French and German governments for political propaganda. For instance, the British had seized the Suez Canal, which gave them military and political advantages, one being they no longer had to sail around Africa but could sail through the canal from India with their troops and goods. Robert Koch had fled from Berlin because he had killed thousands with his magic drug, tuberculin, against tuberculosis. Otto von Bismarck called him back and offered him 100,000 Reichsmark in order to promote the argument that the British would bring in anthrax, smallpox and the plague from India, and thus quarantine was demanded. They were not allowed to dock at any Mediterranean port. This was done to deprive the British of their advantage and also justify shooting at them at Gibraltar. The invisible virus, the terrorist in the air, brought by their enemies. In germ theory, everything is reversed or inverted. The bacteria that keep us alive and support our health are said to cause disease. The word germ, which means new life, new growth, such as germinating seeds, or an egg in our bodies being germinated to bring forth new life, in germ theory means an agent of disease and death. The word isolate to everyone else other than a virologist means to separate from another substance. In virology, it means to add more things. Instead of purifying out a single component to study it, you increase the complexity of the mixture. The method for isolating particles in microbiology has been around a long time. You take the sample or the body fluid, Then you put it through very small filters which separate the tiny particles from the larger bacteria. Then you take the filtrate and use what they call a density gradient ultracentrifuge. You use the test tubes with a density gradient solution such as sugar, putting the more dense ones at the bottom. Then you add your sample and spin it. This separates things by their density. This forms very tight bands that enable you to extract the bands containing particles of the same density. So you have them in a purified state. This is isolation. You separate it from everything else. Then you can look at them under the electron microscope, do chemical characterizations on them, and extract genetic material if you want. You can then do tests to see if what you have is pathogenic or not. Without these steps, you do not have a sample of a virus, and so you cannot prove it caused a disease. You cannot create a diagnostic test, nor a vaccine. Has this ever been done in virology? No. Despite the fact that there are a large number of publications that claim the SARS-CoV-2 virus was isolated, isolation was never done. In fact, the majority of published papers on viruses contradict the claims made in their titles or introductions. The majority of doctors do not have time to read these papers. Most have to see large volumes of patients as quick as they can to make a living. Instead, there are services for doctors such as Medscape, which summarizes selected papers, which they choose of course, with titles and blurbs. So a doctor would assume what is stated in these blurbs is actually what is in the paper. What virologists call isolation was dreamt up by John Enders in 1954 and became the gold standard for virus isolation. John Enders thought that if you are going to grow a virus in a lab, you would have to do it in the same kind of cells that it infects in animals. So for polio, that supposedly infects the spinal cord, you would have to grow it in spinal cord nerve cells. But this was a very difficult thing to do. So he got the idea of using aborted fetal cells that were easier to grow in the lab. He put diseased spinal tissue from polio patients in fetal cell cultures. The breaking down of the cell culture was considered to show the signs of disease. To him, this was proof that a virus was present, despite the fact that he added no virus to the culture. He added chopped up diseased spinal cord. From this culture, he manufactured a series of toxic cell cultures from which the polio vaccine was produced. For this, he received the Nobel Prize. He used the same technique for measles, and this manufacturing process became known as virus isolation. The paper published in 1954 by Enders and Peebles, Propagation in Tissue Cultures of Cytopathogenic Agents from Patients with Measles, which you can find on the internet, describes how patients diagnosed with measles, in which they could never find a virus by the way, were asked to gargle with 10 to 15 milliliters of cow's milk. Specimens from the throats of younger children were obtained by a cotton swab that had been moistened in milk. The swabs were then immersed in 2 milliliters of milk, penicillin and streptomycin, which were added to the samples. Also added was supernatant fluid and sediment, and about 10 milliliters of blood in a solution of heparin. According to Enders, the reason the cells were treated with highly toxic antibiotics was to prevent any bacteria from killing the tissue. So if the tissue dies, it must be a virus, because all the bacteria have been killed. After the addition of antibiotics, feces suspensions were prepared by grinding the material in bovine amniotic fluid medium. Amniotic means the bag and liquid that surrounds an embryo. This is to slow down the dying of the culture. Also ending up the mix was human and monkey kidney cells, beef embryo extract and horse serum,
etc., etc., etc.
After all that, the culture is then starved of nutrients over a number of days. It did not come to Ender's mind that the antibiotics were killing the cells, and it didn't come to his mind that the reduction of 90% of the nutrition that was used to feed the tissue was the cause of the dying tissue. The subsequent dying of the kidney cells was then interpreted as the presence and also isolation of the measles virus. A virologist's excuse for starving and poisoning the tissue would probably be, we are just making the tissue more vulnerable to the virus so we can highlight the virus effects. Enders and Peebles did warn in their paper that additional observations, however, will be required before it can be confidently asserted that dermal epithelial cells are specifically attacked by these viruses. And it must be borne in mind that cytopathic effects which superficially resemble those resulting from infection by the measles agents may possibly be induced by other viral agents present in the monkey kidney tissue or by unknown factors. Despite these warnings, no control experiments were ever performed. The reason virologists do not do control experiments to determine if it is the method used that produces the effect and not the sample from someone they think might have a virus is that it would discredit the whole of virology and upset huge commercial interests. There is a lot of money to be made from vaccines. Stefan Lanker, a German microbiologist, instructed a lab to carry out the control experiment that virologists won't do. On April the 21st, 2021, Dr. Stefan Lanker announced the first results from the experiment. WHO protocols and materials were used to determine if the cells die, known as the cytopathic effect or CPE, when there is no infected sample added that might contain a virus. Only the culture itself is used in the experiment. The tissue culture cells were prepared using full nutrition until they were ready to start the experiment. They reduced the nutrients from 10% to 1% as per virologist protocol, known as a starvation diet. Then they added triple doses of antibiotics, exactly as the virologists do it. By day 1, early damage to the cells was observed. By day 5, full cytopathic effects, dying cells were present, proving that the culture medium itself killed the tissue culture cells. This experiment completely disproves the method used by virology to claim that viruses exist and cause disease. In the picture, you can clearly see that the antibiotics and the removal of nutrients produce the psychopathic effect. This procedure is not just one of the ways in which virologists prove the existence of a virus, it is the only way. So here we have the death of virology. Have you heard about it on the news? No? I wonder why.